Moving on to the pathogenesis of Guillain-Barré syndrome, we will see the immunopathogenesis of GB syndrome. We will talk about the antibodies that are specific to each subtype, and we will see what is the pathophysiology of GB syndrome. If we talk about the immunopathogenesis, we already know that AIDP or acute inflammatory demyelinating polyneuropathy is the most common type of GBS. Cellular and humoral immune mechanisms are both recognized as responsible for tissue damage in this disease. AIDP is closely analogous to an experimental T-cell mediated immunopathy, designated as experimental allergic neuritis or EAN. It is induced in lab animals by immune sensitization against the protein fragments that are derived from peripheral nerve proteins, in particular against the P2 proteins. So based on this analogy to EAN, it was initially thought that AIDP was likely to be primarily a T-cell mediated disorder. But abundant data suggests now that there are autoantibodies against non-protein fragments which are central to many cases. So what is the cellular or cell activation? It is suggested by the finding that elevated levels of cytokines and cytokine receptors are found in serum and in CSF. Those which are found in serum, they include the interleukin-2 or the soluble interleukin-2 receptor, and those found in CSF are the interleukin-6, the tumor necrosis factor alpha, or the interferon gamma. Continuing with the immunopathogenesis, immune responses to non-self antigens misdirect to host nerve tissue through the resemblance of epitope mechanism, which we also call as molecular mimicry. The neural targets are specifically the glycoconjugates, or which are also called the gangliosides. They participate in cell-to-cell interaction between axons and glia, and they are responsible for modulation of receptors and regulation of growth. Gangliosides are exposed on the plasma membranes of cells, so that since they are exposed outside the cell, they are more susceptible to antibody-mediated attack. They are present in large quantity in human nervous tissues, specifically at the nodes of Ranvier. Anti-ganglioside antibodies are most frequently found to GM1. They are common in GB syndrome, mostly found in 20 to 50% of the cases. They are particularly found in the axonal variants, which is the acute motor axonal neuropathy and acute motor sensory axonal neuropathy. And in most of the cases, they are preceded by a Clostridium jejuni infection. Now, this is the postulated immunopathogenesis of GBS, which is associated with Campylobacter jejuni infection. B cells recognize glycoconjugates on Campylobacter jejuni that cross-react with ganglioside, which is present on the Schwann cell surface, and the subjacent peripheral nerve cells. Some B cells activated via a T-cell independent mechanism secrete primarily IgM. Other B cells that are activated via a partially T-cell dependent route, and they secrete primarily IgG. T-cell help is provided by CD4 cells activated locally by the fragments of CJ proteins that are presented on the surface of antigen-presenting cells, APCs. As you can see in this diagram, here are the one of the B cells are producing IgG, and they are recognized by CJ proteins, and then they are activated by which are presented on the surface of antigen-presenting cells. A critical event in the development of GBS is the escape of activated B cells from Peyer's patches into the regional lymph nodes. As you can see here, the activated T cells probably also function to assist in opening of the blood-nerve barrier. and they facilitate penetration of the pathogenic autoantibodies. So the earliest changes in myelin, they consist of edema between myelin lamella and vesicular disruption of the outermost myelin layers. These effects are associated with activation of C5b C9 membrane attack complex and probably mediated by the calcium entry. It is possible that the macrophage cytokine TNF or the tumor necrosis factor also participates in myelin damage. And so this is the immune pathogenesis of postulated immune pathogenesis of GBS that is associated with Campylobacter jejuni infection. Uh, the principal anti-glycolipid antibodies that are implicated in each subtype of GB syndrome are there in acute inflammatory demyelinating polyneuropathy. There are no clear patterns of the antibody target, and the usual isotype that is found is IgG, and it's polyclonal. In case of acute motor axonal neuropathy, GM1 is the most common antibody target. Other than that, there is GD1A, GM1, GM1B, GalNAc, and GD1A. These are found in 50% of the cases of for these other types, antibody targets are found in 50% of the cases of AMAN, and the usual isotype is also IgG, and it's polyclonal. In Miller-Fisher syndrome, the antibody target is GQ1B, and it's found in more than 90% of the cases of MFS. And the usual isotype is also IgG, and it is polyclonal. In acute pharyngeal cervicobrachial neuropathy, GT1A is the antibody target, and the isotype is IgG, that is polyclonal. Anti-GQ1B IgG antibodies. These are found in more than 90% of the MFS patients, and they are not found in other forms of GBS. Unless there is extraocular motor nerve involvement, because these antibodies are related to the extraocular manifestations. Titers of IgG are highest early in the course. Other antigenic targets include IgG antibodies against Schwann cells and neurons that are capable of mediating disease following direct passive transfer to naive hosts. If we talk about the antibodies, there is complement deposition along outer surface of the Schwann cell. There is activation of complement, which leads to the vesicular disintegration of myelin sheath. Whenever there is vesicular disintegration of myelin sheath, there is recruitment of activated macrophages that participate in damage to myelin and axons. What happens in acute motor axonal neuropathy? There is deposition of complement along with IgG in the nervous tissue, specifically at the nodes of Ranvier along large motor axons. Antibodies against GD1A have very fine specificity in AMAN. It favors binding to motor rather than sensory nerve roots. However, ganglioside is expressed on both fiber types. So this is the specificity of AMAN, or it's the characteristic of acute motor axonal neuropathy, that it favors binding to motor nerve roots. These are the structures of antibodies. As you can see in the diagram, this is GM1, this is GMB. These are the different chemical structures which differentiate these antibodies. So, and these are different amino acids which are given, which combine and make these antibodies. If we talk about the pathophysiology of GB syndrome, this is the most common demyelinating form of GBS is the acute inflammatory demyelinating polyneuropathy. What happens is, whenever there is damage to axon, it results in conduction block that is demonstrable electrophysiologically. Axonal connections remain intact after regeneration of the axon. You can see in this diagram there are unidentified antigens which are binding to the myelin, and they are causing damage to the axon. Macrophages are accumulating and they are damaging that part of the axon. Whenever a part of axon is damaged, there will be a conduction block. The conduction won't be possible across that area, and it will not happen unless the axon regenerates itself. These are the GM1 and the GD1A antibodies that are attacking the nodes of Ranvier, and you can see here clearly. This is myelin and these are Schwann cell microvilli. These areas are damaged mostly by the antibodies. Recovery is rapid as remyelination occurs. So as soon as the remyelination occurs, there will be rapid recovery. If we talk about the severe cases of demyelinating GBS, in severe cases there is secondary axonal degeneration. Whenever there is secondary axonal degeneration, the rate of recovery will be slow, and it usually occurs in the axonal variants of the GB syndrome. There is greater degree of residual disability, and severe primary axonal pattern can be demonstrated electrophysiologically. Motor axonal cases, the lesion is localized to pre-terminal motor branches, and regeneration and reinnervation occurs quickly. In mild cases, there is collateral sprouting and reinnervation, and to re-establish physiologic continuity with muscle cells, it takes place over several months. This was all about the immunopathogenesis of GB syndrome. Keep watching this lecture. Thank you.